My name is Sam Chevalier, and I'm a Course 2 graduate student. And on behalf of the planning team which put this event together, I would like to welcome you all to the Veritas Forum at MIT. This forum is sponsored by the Secular Society of MIT, Graduate Christian Fellowship, Reformed University Fellowship, Tech Catholic Community, Baptist Student Fellowship, InterVarsity, Crew, and the Asian Baptist Student Koinonia. Tonight's event is not a debate. We are not here to see people win, lose, or score points off of each other. Veritas is Latin for truth. So we're here tonight for a real discussion, connecting truth to hard questions with our sincerest beliefs. We have asked our presenters to share personally, ask honest questions of each other, and model a conversation that all of us can continue this evening and beyond. Our hope is that everyone in this room would be challenged. Through these forums, we aim to create events that are relevant, thought-provoking, and engaging to people of all backgrounds and beliefs. And we need your help with that. Please pick up your Veritas survey card and the pencil we provided, and take 15 seconds now to fill the top half if you haven't done so already. The top half of the front side, that is. We'll only use your email address to send you a link to an online feedback site where you can share your thoughts on tonight's forum. We really want your feedback, so please do sign up for that. We will not email you about anything else unless you check one of the boxes at the bottom, or unless you win the raffle for the new iPod that we're emailing to let you know about. After the conversation, please fill out the remaining sections of the survey card, both on the front and the back side. At the end of the forum, please bring your card as you exit and leave it in one of the boxes provided. We are thrilled to have tonight's conversation between Dr. William Hurlbut and Dr. Rudolf Jaenisch, moderated by Dr. Cullen Bowie. Dr. Hurlbut received his undergraduate and MD degrees from Stanford University. He then did postdoctoral work in theology and medical ethics, and since 1985, he has acted as a physician and consulting professor of neuroscience at Stanford. During that time, his primary research interests have included the ethical issues related to biomedical technology advancement, including stem cell research, and the integration of theology and the philosophy of biology. Dr. Jaenisch received his MD from the University of Munich, and he is currently a professor of biology here at MIT. In one area of his research, he uses induced pluripotent stem cells to study the genetic basis of human diseases such as Parkinson's, Alzheimer's, autism, and cancer. He is also a founding member of the Whitehead Institute for Biomedical Research, which is one of the world's leading centers for genomic research. Last but not least, Dr. Cullen Bowie, who is serving as our moderator tonight, has received his MS and PhD degrees from Stanford University, and today he is an associate professor of mechanical engineering. His research lives at the intersection of electrochemistry, electrokinetics, and microfluidics, with applications in areas such as material sciences, microbiology, and energy storage. Additionally, he is running a new startup company called Kaitopen that is commercializing his lab's technology to make gene editing techniques like CRISPR faster. We are privileged to have these three people join us for tonight's Veritas Forum. I hope to see all of you at future gatherings exploring other facets of these issues, and I encourage you not to let this conversation end here. Again, thank you all very much for coming tonight, and please join me in giving a warm welcome to our speakers.
Okay, so good evening. Welcome here. So what I understand is my role is here to give you a scientific basis of gene editing. So we know what we're talking about, and we're not talking about things which may be not quite reflecting the reality. So let's put you on stage. We are composed of 22 pairs of chromosomes, 2 sex chromosomes, 6 billion base pairs. We have every 1,000 base pairs, we have a mutation. Most of those come from father and mother or mother, but we acquire mutations. And this leads to the variation between, for example, height. So we are in chromosomes, our genomes in chromosomes, and eventually in DNA, and eventually in these 6 billion bases. And if you, let's say, have here a C to T transition, C and T to two of the 4 bases, then let's say you're predestined to get Alzheimer's. If you have, let's say, This one, A to T, no phenotype, it's silent. And if you have this one, you're getting cancer. So genetics is really important for your phenotype. So the question we're going to debating today is really, is it possible to correct such mutations and the potential to treat the disease? And should this be used in medicine? And what would be the purpose? So these are the key questions I want to concentrate on. But we have to talk a little bit about the technology, because the key technology is gene targeting. And I have to go a little bit in the history with 2 slides to put this in perspective. So in the 80s, homologous recombination was invented by Marie Capecchi, where you have, I don't have a pointer, I'm afraid, where you have a piece of DNA, like this one, which you want to insert into an endogenous gene. What you do is you build a vector which has homology, in this blue here, to the sides of where you want to insert it. And this then is a crossover which inserts the DNA into the endogenous gene. Homologous recombination revolutionized biology in the 80s. And what you can do is, the event you're looking at is a very rare event. You have embryonic stem cells, and I assume you know what embryonic stem cells are. They're pluripotent cells. They're really very important. You do your manipulation, and you get one, let's say, 1 in a million cells, which has the expected and desired modification. But you can clone these cells, make a whole dish full of them, and now you want to make a mouse. What you do is you inject these cells into an early embryo, into a blastocyst, and then they can integrate in the embryo, contribute to all tissues, and make what's called chimeric mice, where the white is from the cells, from the modified cells, and this would be in a foster mother. And then, if you're lucky, it contributes to the germline when you cross these 2 mice. So it's a way to convert a cell, which is mutant, into an animal. So what is the issue here? Gene targeting works very well in ES cells, but only in ES cells. It's very inefficient. You need cells to find your very rare event. And it's very time-consuming. This experiment takes, if you're skilled, 1 year. If you're not so skilled, more than 2 years. Long time. So the problem, of course, is for human ES cells, it doesn't work. Or it works very inefficiently. And that was then the emergence of the CRISPR. So I will not go into the history of this. I will just tell you with 2 slides what CRISPR is. So the age of CRISPR. It is the following. You have an RNA, which is 20 base pairs long, which scans the genome for something homology and finds it. And this is associated with a protein. It's called Cas9. And Cas9 is a nuclease. So when these 2 find their target, all what it does is it makes a double-strand break in the DNA. That's all what CRISPR-Cas2 does, a double-strand break. Okay? So there are 2 components. A guide RNA searches for the homology for your target gene, and the Cas9 protein, which cleaves the DNA. So it's basically a molecular scissor. And it really represents a revolution for biology and for medicine. And I will give you some examples for this. And it even hasn't escaped Hollywood, that this is something which could be useful for some story of terrorism or whatever. So CRISPR, you have the Cas9 component and then the RNA. And when it cleaves, you can insert a piece of DNA at the cleavage site. It's reasonably efficient. Or if you don't give a vector, a template DNA, then you can make a mutation, right? So this is the basics. So the question is, how efficient is that? I told you, homologous recombination was very inefficient. You needed embryonic stem cells. How efficient is that? And that's something we were interested in. I'll give you just a few examples. So when you take embryonic stem cells and want to ask the question, how many genes can you target at the same time? With one, and we use in this case 5 genes. And ask the question how many we can do. And the result was 50% of the clones had all 5 genes mutated in both alleles. incredibly efficient. We were really scratching our head. But if you want to make a mouse out of this, you have to go through this long period of injecting the cells and do a blastocyst, make a chimera, a year at least. So can you do it efficiently? And that's a key experiment for what we're talking about today. So can we do it in the embryo itself? Just putting this into the fertilized egg. So in this case, we put 2 guide RNAs for 2 genes, plus Cas9, into this embryo and ask the question, how many of the pups we get are mutant for the 2 genes? And the result was a bit shocking. 80% of the pups had both alleles of both genes mutated. So we made the same mutation the old way in ES cells. It took us 2 years. This took us exactly 3 weeks, the gestation period of the mouse. But what you get there is really, you can't predict. You get some deletion, you don't know exactly how big it is. Can you make a defined point mutation? Exchange 1 base pair. So we did the same guide RNAs, put them into the fertilized egg plus Cas9, plus an oligo, small piece of DNA, 30 base pairs long, had 1.1 base change. And asked the question, can we put this base change into the genes, into the 2 genes? And the answer was yes. 60% of the pups had this base pair change in the 2 genes. So this was amazingly efficient. And this is the basis for what we're talking about. It is efficient. You don't need selection. You don't need screening. It works. OK. So so CRISPR-mediated gene targeting in germline, as I said, it's so efficient. It's rapid, 3 weeks, and you have the mouse, because that's a gestation. So now the question is that useful for gene therapy or of severe diseases, for example, as we discussed? So the applications of this technology is in disease modeling, so people can make not only mutant mice, but also, for example, monkeys. So people have made now monkeys which have a specific mutation which corresponds to major human disease, autism, for example, also. So there would be models much better than mice, of course, to work with. And it is already goes into clinical trials in combination with, as a targeted therapy for certain diseases like sickle cell anemia and whatever. So it is already here. But what we're talking about today, should we use this, or at least what I think is the most important issue, should we use this technology to alter the human germline, to manipulate human embryos? Okay, so that's what I want to now address. So it's efficient, as I said. It is rapid. The key thing, it doesn't need screening. It is so efficient that you can do it directly in the embryo. So should we use this to edit the human germline? So what are the ways we can think about therapy, gene therapy? You can do it on a somatic level. Somatic therapy, correcting mutation, either in vivo of a patient or ex vivo in bone marrow cells which you take out of the patient. The consequences of this manipulation are only for the patient. The patient can give consent. Germline therapy, it's a correction of disease-causing mutation in germ cells, in sperm, egg, or embryo, which has consequences for the next generation. So I have a very nice example of that. Time I can talk about somatic therapy, but I think we'll concentrate on this part because that's probably the most interesting one for this discussion here. So why would you do that? And I give you 3 reasons. Correction of a disease-causing mutation, inactivation of the gene which makes you susceptible to some disease or gives you resistant to some infection, or for enhancement. So let's say these are the 3 general areas one could think about. So I want to give you now in the next 3 slides briefly where the issues are, the problems in using that, as I can see it. And I will first talk about the scientific issues. That's the basis. And I'll later come to some other issues briefly. So correction of a disease-causing mutation. The complicating issues are, if you have a serious disease such as Huntington's, it's an awful disease, it's dominant. If you have this mutation, you get Huntington's disease. There's nothing you can do. It's very terrible. So when you have a parent who is heterozygous for huntingtin. 50% of his embryos will be mutant, 50% will be wild type. There's no way for you to distinguish those two. There's no way. And that's principle, because there's one DNA and you want to modify the DNA. So therefore, you will manipulate 50% of wild-type embryos. And I think that's a problem. And you cannot, and I can go later into this, you cannot assess success because of mosaicism. I will not define this now. Disease-resistant genes, people think about, for example, you could inactivate the HIV receptor. So you would have a baby and then an adult who is resistant to HIV infection. It's used in the clinic now for patients. So you could eliminate the virus infection, or you can correct other mutations to protect you against heart disease. Now, one has to think about here, the alternative, of course, is you can do this also in the patient. So it's done in HIV patients. They're corrected. You delete the receptor, and they're becoming therapied. So you may not need germline requirement for the germline editing. And finally, enhancement. For example, you can insert, it's pretty trivial, insert the growth hormone gene into some expression locus. You will get a taller baby, but I think, so that would be clear, the expectation. And of course, this is scientifically feasible, but I think it's a different issue, which we have to discuss. It's really much, it poses other than scientific issues. So, one thing I want to say, these are the scientific issues which I think argue against editing in zygotes, because you can't assess what you did. You cannot. So it has to be so efficient that you can do it, but biology is never that efficient. But can you overcome that? And that's getting interesting now. If you do it postnatally, can you do it in spermatogonial stem cells and oocyte stem cells? The advantage is those cells can be cloned, and you can assess what you did. And I give you the example. This is a study from Ralph Brinster in the 90s. So he isolates from testes spermatogonial stem cells. They can grow in a culture dish. And when you put them into a testis, they can make sperm. And you can see the blue mice, they can give rise to offspring. The key is, since you can clone these cells, you can manipulate them, have a sister clone, and evaluate whether it was right or not. That would solve the scientific issues. And there's also the idea that you can do this possibly with making oocytes from stem cells, from pluripotent stem cells, oocyte stem cells. So you could do this also with female cells. So the use of these type of cells would may resolve these scientific issues I raised. So now it becomes more interesting. If you would have resolved these scientific issues, should you do it? So, and I give you just a few thoughts about it. I'm sure that Bill will much more go into this. So the ethical worries would be mistargeting causes uncertainty or unwanted problems, aspect of eugenics, slippery slope to eugenics. Once introduced, you cannot really, may not reverse it. Editing may accelerate inequality, and so on and so forth. Arguments against germline manipulation would be intergenerational consent is not feasible, impossible to predict consequences, and the threat to human dignity. The counter-arguments would be intergenerational consent is not relevant for multiple other decisions regarding future decisions. We let our next generation grow up in a climate change world, right? We don't ask them. So I think, so that's one counter-argument. Impossible to predict the consequences for other well-intentioned efforts to improve human conditions. All the time we have that. And I think there's no shared conception, as far as I can see, regarding the notion of human dignity. So I think this is a debate which is ongoing. And let me just say, I was on the committee from the National Academy of Science, which worked out some guidance for germline editing. And the book was published last year, and I just give you a few highlights of this, and I think Bill will much more go into this. So they say, yes, there should be absence of any other alternative. Then you might consider that. The restriction is really you have to be convincingly have the science done, very good science, basically the same. Maximum transparency, it's really absolutely clear what it should be, and reliable oversight. So this is just a very simple summary. So let me come to my final slide. So why should you do this? So disease prevention, Tay-Sachs, Huntington's, I mentioned, sickle cell, that could be you eliminate these disease genes from the next generation. But you should consider the alternatives. I mean, you can, if you don't want a Huntington's baby, if you could make pre-implantation genetic diagnosis and select the embryo which doesn't have the mutation. Very safe, and it's used in the clinic. And somatic therapy, as I said. But there may be no alternative whatsoever. Because, for example, if you're homozygous, if the parents are homozygous for, let's say, Huntington's, or both parents are homozygous for a recessive disease, there's no way they can have a genetically normal child. So this might be the only possibility. And if there's infertility with this Y chromosome linked, will be similar. So that's disease prevention, modifying disease risk genes. So could you, as I said, could you make babies who would be resistant to virus infection by inactivating the HIV receptor, trivial scientifically, or all the other genes to affect your incidence of Alzheimer's or heart disease or cancer. Enhancement, should you do more muscle in your offspring, eye color, learning, memory, and of course, these are very complex traits. So these are the issues you can consider. And I think the point which we probably today discuss, what is permissible, what's not permissible. This is not obviously not a scientific issue. That's something which society would have to decide. And I think I let Bill follow up this. And so I'm not sure if there are questions now or should be later or what.
We'll do them later, but thanks.
Okay.
Yeah.
All right.
I'm always amazed when the audiovisual actually does work. So that was a very good introductory framing of the science. I want to talk with you about the ethical, the social, and sort of personal issues of this. This is an extremely important issue. I can't tell you how significant I think this is, and I think what Rudy's told you already confirms that. About a year ago, Dr. Jaenisch and I were on we're both speakers at a conference at Harvard, and this was the poster, and I thought it was an interesting way to start, to reflect on human hands, what Aristotle called the tool of tools, the symbol of our distinctive body form and our unique capacities of mind, our comprehensive, creative, and controlling capacities as human beings. Really, we're a remarkable species. These hands are now reaching into the most fundamental forces of living nature, and that raises enormous problems. 70 years ago, Aldous Huxley, anticipating the transformation of human life through advances in biology as the final and most searching revolution, asserted this really revolutionary revolution is to be achieved not in the external world, but in the souls and flesh of human beings. Now, in what MIT Tech Review has called the biggest biotech discovery of the century, we have a tool that is finally, after about 50 years of doing this kind of science, it's very precise, very inexpensive, and very quick to do. So we're right at the edge of a major revolution in biotechnology. It's been characterized, and Rudy showed this slide, it's been characterized as a molecular scalpel, but in fact it's really more like a Swiss Army knife. It's got multiple ways of intervening at all levels of genomic process. Together with our exponential increase in gene sequencing capacities, our deepening knowledge of genetics, cytology, and developmental biology, the scope and versatility of this technology promises transformational impact as great, I think we'll see in retrospect, as great as the discoveries in electricity, synthetic chemistry in the end of the 19th century, and then nuclear physics in the early 20th century. This is a dramatic moment, I believe. Physics, chemistry, and now biology, biotechnology, full circle on the Enlightenment dream of mastery and as masters and possessors of nature. We may be finally approaching the revolution anticipated by Huxley, and with it, a huge, profound, and vexing questions about our aims and our applications of this, and the role within, the human role within the natural order, and the proper use of this technology in shaping the human future. At the most fundamental level, these questions go beyond issues of individual rights and social responsibilities to considerations of the very source and significance of the natural world, its integrated and interdependent processes, and the way these provide the foundational frame for human existence, human identity, because it's not just medical issues are going to be addressed. We're potentially going to transform vast swaths of the natural world. The largest lens of these considerations is the natural and social environment that sets the frame for the physical, psychological, and moral and spiritual meaning of human life. In other words, we're about to transform the world of living creatures the way we have largely done with the inanimate world around us. It's a very, very remarkable moment. Is the natural world simply a coincidence within a chaos, an aimless and arbitrary product of physiochemical processes? Or is it a moral and spiritual order requiring humility and a sense of limited human prerogative, of rights and responsibilities that we have to take seriously? Proposals for the use of CRISPR and other gene editing technologies range from the fascinating to the frivolous. Projects of broad ecological engineering, de-extinction of human ancestral species have been proposed. Recombinant actions on animals. We already have a bunny that glows in the dark. That probably didn't hurt the bunny at all, but it's just a phosphorescent gene. But you can imagine with this technology, we might really get pretty wild. And who has rights about that? Do the commercial developers have absolute rights, or is there something about human role over the animal creation that we ought to take seriously? Likewise, there are serious proposals for chimera creatures. This is really a joke, but think about the way creatures could be blended and genes could be blended between species. And then, of course, there's visions of the human future, including engineering the perfect astronaut, treating aging as though it's a disease and therefore reversing it or countering it somehow. There's the astronaut picture. And then there's the transhumanists. Some argue that it's human destiny, it's human nature to just go forward in this kind of technology, to improve ourselves even at the most fundamental levels of our genes. These include, especially the transhumanists. I'm sure you have transhumanists here at MIT. They're an international intellectual and cultural movement advocating technologically mediated enhancement of human intellectual, physical, and psychological capacities. Their logo is H+. It's, human with a plus. I've had several of the leaders of the Stanford Transhumanist Society in my classes. They're intelligent, serious-minded students. They argue that our advancing technology offers us the opportunity to escape the constraints and cruelties of an amoral evolutionary process and the opportunity to lift humanity in its next level of personal and social flourishing to enhancement of human-machine hybrids to the creation of post-humans, they argue. Others with more modest goals, there are some who suggest we should simply use it to make ourselves smaller so we don't consume so much resource in the world, but I think that's really a joke. But others with more modest goals point to the very serious role of these technologies in addressing disease, doing biomedical research, and the final issue that's overarching all of those is the question of our role, our vision of nature, what's nature for, and what's it what's the right thing to do? So I just want to take a quick, way too quick tour of what the human dimension of what Rudy has laid out. This is Jennifer Doudna, who is one of the discoverers of CRISPR-Cas9. Jennifer and I are working on a project together on the social and ethical issues implied by these advances. And Dr. Jaenisch is part of our working group. Very important issues, I think, issues for the best scientists, scholars, and thinkers in the world, but also for the whole human family, because these are species issues. They're not issues for elite, educated people only. They're issues for the whole human family, the people who do the perennial tasks of having the children and the workaday tasks of life. But we all know that fundamental moral imperative of human life is to treat disease, in this case, sickle cell anemia. There's now proposals for taking the cells out, the stem cells out, modifying them and returning them into the bloodstream to repopulate the marrow. A hopeful process, I think it's likely to be effective in the next few years. There's even ways of sending these vectors carrying these agents that Rudy talked about, using viruses or nanoparticles. Gold nanoparticles are the latest thing, liposomes, ways of delivering them internally to the body. Harder, but maybe feasible. And then so we get to the point where we ask ourselves, well, what about it? What's the right thing to do? And as Dr. Jaenisch pointed out, there's somatic cell interventions and germline interventions. But at either level, we can feel the imperative. This poor little fellow, and this is a disease called Lesch-Nyhan's disease, where the children actually chew their fingers off. It's missing a certain metabolic factor, and it's very terrible, and you feel the weight of this. But what's right to do, and how much risk, and is it really right for us to start engineering the human genome? And then you ask yourself, well, where will it go? It's not just very serious diseases, but what we might think of as quasi-conditions. This is albinism. Is it really a disease or a human variation? Well, you could argue that there's deficiencies with it, but people live very good lives with albinism too. And then you blend over to the obvious issues of parental preferences and eye color, height, and so forth. Not going to be easy to do, by the way, any of that. But still, it raises issues of enhancement. And when you start to think about all the things that are genetically grounded, it really raises a lot of possibilities. Although, let's not be so foolish as to think genes control everything, because even identical twins are not all that similar when you get down to the actual processes of their biology. So what would we do? Well, are there good intermediate things to do? This is George Church from Harvard. George has identified what he calls rare protective alleles. These are dimensions of found in the human gene pool that are variants with large impact that exist naturally, but they're not as common as they could be, and may be beneficial if more people had them. There's a whole range of these, and George has suggested that we might want to improve the human stock by doing, there's a whole bunch of them with significant things. Of course, number 4 is variant of low odor. I'm not quite sure what that's about, but in any case, maybe that'd be the most popular, I don't know. But it does raise challenging questions as to whether it's our role as human beings to improve the human gene pool. But other people, like developmental biologist Stuart Newman, warn that the genetic design of future offspring, even limited objectives, make it difficult because there are different definitions of normality, different access to technologies, and different willingness to take risks with future lives, which is a serious bunch of considerations. But where I come from, and I'm sure it's true right here too, there's a spirit of innovation and quest and discovery. So there's a lot of momentum behind the idea of improving human beings. And yet if we go back a ways into the history of our species, we realize that there's a long history of considering what the role of human beings is in their own interventions against disease. The Roman physician Galen said that the physician is only nature's assistant. But now look at what we are considering doing. Now with the powers of our advancing technology, There is new paradigm, one of liberation, technological transformation in the quest for happiness and human perfection, grounded in the widespread practice and general acceptance now of cosmetic surgery and other lifestyle interventions by using pharmaceuticals. Slowly but steadily, the scope and purpose of medicine are being extended along the gradient of our appetites and ambitions to encompass dimensions of life not previously considered matters of health, but natural human variations. And that raises a very profound question. I mean, look at the things we've done. Growth hormone, those people were not really, they don't exactly have a disease, they function well, but they're not socially the right size. And then people have accepted birth control pills, Viagra, lots of interventions. We're getting to the point where we're trying to remake our lives. And when you think about it, who wouldn't want to have certain advantages in life and certain advantages for their own children? So there are proposals for better brains, including, by the way, proposals for intervening in genetics in a way that would produce more moral human beings. I'm not so sure that's even possible, but it's been proposed, advocated. And of course, there's quite a thrust in California. Google has the Calico, the California Life Institute, working on longevity interventions. And I was at a conference where William Hazelton said the real goal of biotechnology is to keep people alive forever. And it's natural. Human beings want perfection. They want for themselves and their children. So this is a huge temptation. When I served on the President's Council on Bioethics, we produced this volume that examined these issues and looked into the underlying questions, and it was not easy to define the difference between therapy and enhancement. This, for example, was considered a therapy at the time before the Civil War in the antebellum South. This was a defined disease in the textbooks. It was called drapetomania, from the Greek words, a tendency to run away, a passion to run away. And you can see his treatment as a slave was to be whipped. And of course, we all know the sad history of Nazi Germany, which was trying to aim for perfection, but ended up terribly tragically. So it's not easy to define a disease, and it's not easy to tell people what they should and shouldn't do. Of course, everybody knows about Lance Armstrong, and so we have a precedent for this. And I'm not trying to exaggerate this, it's going to be difficult, but it actually is no longer completely science fiction. We're moving into the realm of technologically altered human life, raises profound questions, and those questions were raised and addressed initially through Jennifer Doudna's concerns. There was a gene editing summit that Rudy referenced, and they were taken seriously. But at the same time, they pointed out that to address complex diseases like cancer, we must carry our investigations to the most fundamental elements of living systems, which brings us to the most profound questions, besides the modification of human embryos for therapy, is obvious use of modification of human embryos for inquiring into the very nature of developmental biology. So before we had these multiple embryos, and there was controversy over the development of embryonic stem cells, but now there's a vast new arena of possibilities for the use of embryos to study specific disease mutations in embryos to produce embryonic stem cells that could serve as models. So now the tools are in place for vast increases of valuable science for sure, but controversial science. So remember when the stem cell issue our country was divided deeply about this. And in my own medical school had the cover of our alumni magazine pitted science against religion. That was a false dichotomy. There were many people on both sides who were religious or against embryonic destruction for non-religious reasons. But both sides had their arguments. Good science could come. On the other hand, there was the defense of human life as a living human organism from its initiation to the end of its natural life with death. This is an 8-cell human embryo on the tip of a pin. And the argument was, well, human beings at all phases of life are a developing individual organism, and there's a continuity of being, and that therefore we should defend human embryos as the sanctity we give to normal human life at other stages. This was a very, very difficult controversy that we all worked through, sort of, but never really resolved. But it did leave us with what I want to address as the final issue here. If in this age, there are many uses we could make of human embryos with this new technology, and we're going to have to decide what our principles are. I just want to raise 4 questions. Will we now endorse the use of human embryos for a wide range of studies of infertility and early development, not just for getting stem cells? There are many, many ways to do this, and many useful experiments could be done. Second, will we allow the creation of embryos specifically for research purposes? They do allow that in the UK, by the way. And I had that in there from past lectures, so I thought I'd leave it. That's Dr. Jaenisch. Is that the very mouse, Rudy, that was created? He made the first transgenic mouse. In this case, he was telling me at dinner, it wasn't exactly a study of developmental biology, but you can see how if you could knock genes in, knock genes out, we could study human embryos and understand human development the way we've studied mice for the last 30 years. So there's no doubt that the science could be very useful. The question is, how about the ethics? And if we do this, how many embryos would it be okay to use in research? Well, a Japanese scientist created 581 copies through rounds of cloning. And whether it's cloning or some other method, creating batches of identical embryos and doing controlled studies would be a very useful scientific enterprise. How would we get the cells to make those embryos? As Dr. Jaenisch pointed out, we're getting to the point of harvesting them through in vitro production from induced pluripotent stem cells. But there might be other ways too. But 15 years ago, a stem cell biologist named Dawo Salter raised this issue. He said you could have millions of gametes, you could combine them, and today we can't do these experiments on human embryos because it's considered morally repugnant, they're difficult to get. But if embryos could be grown in culture like any other cell line, this latter problem would disappear. They'd be easy to get. They'd become like any other cell type. They would become objects and would be used as objects. And then he went on to say, I have no idea what kind of moral value or rights we would give these embryos. We'll probably go through the same agonizing we did with IVF. It could be terribly difficult to begin with, but then would become a fact of life. 20, 30 years from now, we might read in the papers somebody made 20,000 embryos, studied their development, and we'll decide it's okay. So you can see huge issues. And then, of course, the very, very difficult one is, will we allow research on embryos beyond 14 days? And if so, for how long? And according to what principles of moral valuations? In the United Kingdom, they allow embryo research up to 14 days. But now that they can actually keep them alive longer than 14, there's a huge controversy going on about whether they should revise that rule and allow it to go on. There was just an article in Nature Biotechnology 2 months ago about this big debate going on. And this scientist at the Francis Crick Institute is already doing studies of developmental biology with human embryos. So there's going to be a big controversy over this, probably an international controversy, and it's going to be difficult. Now, would anybody really want to go beyond 14 days? Well, my colleague at Stanford was on the California State Commission on Human Cloning, and somebody asked him why they drew the line at 14 days. And he said, oh, that's just the first limit, just so people get used to it. And we could change our ideas about how long in the future based on likely changes in our understanding of neuroscience. Well, what does neuroscience have to do with it? And of course, what's gravitating slowly but surely is the notion that you're a human being, and only when you're conscious, self-reflective, and so forth. So what I'm raising this for is because this is going to be another very difficult debate. And would anybody do this? Well, I debated this guy, Julian Salvalescu, some years ago at a forum in New York City, and he said that not only would it be okay to do it, we have it morally required to produce clones, implant them, and harvest them for their cells, tissues, or even organs. I asked him, how long would you let it gestate before you harvest it? This guy's a physician, by the way, and head of a bioethics think tank at Oxford. And he said, I said, 6 months? He said, yes, I'd go 6 months. So there's going to be controversy. And Erwin Chargaff, who was a major figure in early genetic technologies, warned us that we're going to have to face these profound questions as biotechnology advances. He warned us that the time was coming when there would be useful cells, tissues, and possibly even organs that could be extracted from human beings, early human beings, like we currently take, like at one point they extracted gold teeth. So I don't want to sound pessimistic about that. I don't know any scientists that have terrible scenarios except for a few that are like that guy, Julian Savulescu. But nonetheless, there are different cultures and different nations. And I gave a talk in China a year ago at the 60th anniversary of Renmin University, which is the original communist university. And I was talking about these kind of issues. And afterwards, the chief ethicist, bioethicist of China came up to me and he said, you know, he was commentating, came up and was commenting in front of the audience. And he said, you know, in China, we don't believe you're a human being until birth, and therefore those kind of projects would be okay. And so he said, Confucius says you're not a human being until birth. Afterwards, a philosophy professor told me Confucius does not say that. But be that as it may, we've got difficult international issues to contend with. So what is the connection between human beings? That's the only slide I can find that says man, but it's obviously humanity. What are our roles? What's our prerogative? And how can we engage the people who do the normal human life that don't necessarily have scientific perspectives, but have strong and beautiful opinions about what's good and true in life? The people who work hard, sacrifice for their children, and hold our society together, they need to be engaged. So I just want to end with a picture of hands again. What is our place as human beings? Obviously, this is from the Sistine Chapel, and it raises a profound question of what are we? Are we co-creators in the world? And if so, in what sense do we operate in the realm of both science and spiritual truth to do the good that we are capable of understanding? Profound creation we're part of. It's mysteriously beautiful and horrifying at once. There's tragic disease, but there's also very moving dimensions of evident beauty, goodness in the natural world. So how do we find this mysterious balance? And how do we go forward in a way that is humble? The very word humanity comes from the same root as the Latin word humus, earth or soil. And therefore, it's the same root also of the word human. So human beings should be humble. We're creatures of the earth. We're formed and fashioned by the earth, and we have to be humble as we dwell in it. And we should never let ourselves, for sure, we should never let ourselves get carried away by our ambitions and appetites and forget the profound needs there are at the very core of humanity. Thank you very much.
Okay, am I on? We good? All right, so thanks so much for both of you for your words. And I think one thing that stood out to me was there are some words that come up that are difficult, that are kind of germane to this discussion, that are difficult to define, things like good and severe and disease. And I think before we dive in a little bit further, a question I have for you is kind of when I think of like, what is a good life? What constitutes a good life? I think to define what is a good life, you have to know, well, what is your worldview? What defines a good life? And so I'd love to hear both of you talk about your view of the world, perhaps faith, religion, or otherwise, and how that influences your, if at all, how you think about these topics.
You on, Rudy? Up to you.
Colin, my worldview is that of a practicing Christian. I was not raised in a religious family. I was kind of a radical student in the late 60s at Stanford, and then I became dissatisfied and frustrated by what was going on in the culture I was part of. And I went back to my room and I had a Bible I'd been given because my parents forced me out the door to Sunday school. I didn't want to go, but they didn't go. So I had a Bible that had been given to me when I was confirmed. And I really didn't understand the first thing about it, but I decided I'd read it. And I think maybe I was okay with it because I remember Jesus had long hair. So, that was the 60s.
I don't know if that was in there.
That wasn't in there.
Yeah, that might have been the pictures.
Yeah, that's like that joke about the kid that goes to his father and says, I want a car. And his father said, okay, you get straight A's, you read the Bible every day, and you cut your hair. This is the 60s, long hair, and the parents didn't like it. So 6 months later, he comes back, and he says, okay, Dad, here's my report card, straight A's, and I read the Bible every day. And his father said, he said, so where's my car? And his father said, well, what about your hair? And he said, well, Jesus had long hair. And his father said, well, Jesus walked. So I became a Christian reading the Bible, and it shaped my life profoundly. I don't claim any single denominational emphasis. I just don't understand the arguments well enough. But I do define my sense of truth and goodness around the life of Jesus, and I don't just mean ethically. I mean in the very deepest sense that I believe that in the higher cosmology implied by the phenomenon of the created order and the destiny of the created order that's spoken of there. So that shapes my life. It is not, however, the only input. I use my scientific thinking and my logic as a human being, drawing on just plain reason, to try and think through where some things are beneficial and good, and so it's a worldview that's grounded in a certain spiritual metaphysics, but also in just normal living and human understanding.
Okay.
Well, okay, it's very different. I was baptized, but I left the church as soon as I could make my own decision. For me, the important thing is I want to find the truth of the world, and the concept of God doesn't help me to do that. So therefore, I do not believe in something like what many of you do, and I don't think this affects my being ethical or being not a bad person, whatever you want to define it. It's not relevant for me to have that as a important background to do that. So we can talk about this, why this is, but maybe that's not the right thing here to do. But what I think, and you asked me that question before, why do you want to talk about ethics of science, let's say, we go back to our theme. I think the science we have now, and we talked this evening about this, is in contrast to the science I did 20 or 30 years ago, which was very far away from any application. You would write in your grant application that all has importance for human diseases. I never believed that. I think it was just a phrase. But now I believe it. It's very close. So obviously you have now the obligation to really be transparent, to know, to tell people that's what's going to happen if you do that. And so I think there's an obligation anyone who works in science has to be transparent and to be informing people. And certainly Bill and I have done this in some occasions. The question, what should you do as scientists when you design an experiment? If the experiment could have bad consequences should you not do it, if it's fundamental research. And I believe this is not the right approach. You cannot forbid people to think and to explore. I think that's, to me, it's almost anti-human. Everything can be misused, everything can be used in the right way. I think where the border is, and it's a very strong one, when you apply it to human beings, to medicine. This is not a scientific basis to this, but it's a different type of decision. And I think that's where, very clearly put the border. This is a decision we as society, we as human beings have to make. Do we want that or do we not want that? There will be very many different opinions, and you voiced some of those. The transhumans and people who, I was being on this committee from the National Academy of Science. For me, it was a very strange realization that people who are deaf don't consider this as being a handicap, but as being a cultural trait. So they won't get deaf children. And there was this case where this couple looked for sperm donors who had a high chance to give them a deaf child. Now you see, they're very far away views on how to use this technology. So I think that's what we'll have to figure out, how we come to a consensus, if there's a consensus possible. And maybe this discussion is an attempt to do that.
What do you think of that, Rudy, creating a child intentionally deaf?
I think it's totally, to me it's crazy, right? It's totally unacceptable. Of course, and one can ask the question, can you make decisions on the next generation? As we said, when you edit and change a human embryo for whatever reason, it's a decision you make of someone who can't give a consent. So you have to think about that. Right? So I think there are very complex questions, which I'm unable to answer. I have an opinion on those, but I can't answer them.
So you mentioned drawing a line at when these technologies affect humans.
Yes.
And Bill, in your talk, you discussed there's differing views on when does human life start. And so I guess I'm curious in hearing you guys discuss a little bit around that issue. So when do we have a human? When do we go from cells to a human? So
I can give you when human life starts. As a biologist, there's no other answer than with fertilization. Then life starts. I mean, this is not the question which is interesting. The question is interesting. When do you consider a human embryo as being worth to be protected against whatever? That's, I think, is the key question, not when life starts. I find this a wrong question. Fertilization, what else can you say? But this doesn't mean that the fertilized egg is untouchable. To me, that is, and I think the compromises we have made, the Britons had made as a first country, that you can use fertilized human embryos to make embryonic stem cells, I fully support, with the clear-cut restriction they had. So that was after life began, of course. You had an embryonic stem cell, you had a blastocyst, which you after 7 days put in a culture dish, you make an embryonic stem cells. So I find this the right decision the British did first. And we have done it in my laboratory. We have generated human embryonic stem cells under the right conditions. And I don't see that this is something which I find problematic for me.
What's your perspective?
Rudy, let me just ask you about that. How do you feel about the controversy in England to carry it beyond 14 days?
So I think I would differentiate a lot. If it's 14 days beyond implantation, right? So the British made a very clear rule. You can do something with a cleavage embryo, with the embryo which grows in a culture dish, up to 14 days. And then you have to make an embryonic stem cells. But once the embryo is implanted, so implantation is a red line. Once it's implanted, you can't touch it. And I would think that is really a good line because there's no gray zone. Either you implant or you don't implant. There's nothing half, right? So now comes the problem with embryos, human embryos being grown in a culture dish beyond 14 days. And there's no experiments published. You can make use embryonic stem cells or mouse embryos, which you can grow in a culture dish up to the primitive streak stage. So when they do get an axis, have symmetry, and get a beating heart, that can be done outside of the uterus. No. So this is, of course, it's a very new development. So this line, the British line, might eventually be getting a little more fuzzy, right? I agree with that. You can make these human embryos, embryo-like constructs from embryonic stem cells, which, of course, it doesn't involve an embryo, but you get an embryo-like structure in culture. Now, is that different from when you make the same structure from an embryo than from embryonic stem cells? I wonder. So these are, I think, interesting issues. For me, manipulation of an embryo in utero, for whatever reason, if it's therapeutic, I would support it. If it is for research or for manipulating, I think then it's a really very complex, and I would have a lot of hesitation.
So if you could develop an artificial endometrium in a lab and implant the embryo and grow it to 20 or 30 days?
I think it raises very complex issues, very complex issues.
So one thing I didn't put in my presentation because I already talked too long, but George Church at Harvard has published a paper on what he called sheaths, synthetic human-like entities, embryo-like entities. And he says that the 14-day rule was interesting when it was just going from the beginning and working up through natural development. But now with the new tools of developmental biology, you can create later cell types, join them, and they self-organize to the level you can actually get cerebral tissues and brain tissues. And he's asking the question, which doesn't really have an easy answer, when does this have some kind of intrinsic dignity? Because most people do believe that what endows human life with dignity has to do with something to do with our neurologic system, obviously. And the question is, well, could we create anything in the dish even if we didn't come up from the bottom, but just, bypass the early stages and create partial human parts of humans in dishes? And I must say I doubt that a cerebral organoid, that's what they call them, just brain tissue would have any kind of consciousness, or it doesn't seem true to the nature of biology to me, but nonetheless it does raise profound questions because we don't actually know what consciousness is. I mean, if you're honest about it, nobody knows. We don't know what it arises in, and most of us in science suspect that it's a molecular mediated phenomenon at the very least, and yet we don't know the minimum construction of it. And so it's just, we're looking at a very, very complex future dilemmas from all this. And
yeah, I mean, I agree. I mean, organoids, you grow in a culture, they will never think, right? These mini brains or whatever I agree. But I think it's interesting to do this in vivo, make chimeras. So make human mouse chimeras, which we're doing for certain questions, because we want to study human disease in an in vivo context. I don't have any problem with this because it will be never human. But you go on, if you make human monkey chimeras, then it gets very complex, right? That's something I think will not be allowed, because then you don't know what you have, because it will be the integration of the human cells will be high, in mouse it's very low. They're evolutionary too distant, but in monkeys they're close. So what is it? Something between a human and monkey. So then I think you cross the line, and I don't think this will be allowed. But
well, I think one challenge is the line differs for everyone.
Yeah.
And I think even if we were to discuss for hours, we probably may not come to a consensus on where lines are. But one line I'm curious in is how would you both define medical use or therapy versus enhancement? So for example, you mentioned the example earlier of a couple that was deaf and was interested in having a child that was also deaf. And that might sound crazy to us, but that's really our opinion. And is our opinion any more valid than that couple's opinion in a decision like that. So how do we come to consensus on these challenging issues when people can have differing opinions on what is valuable and what is good?
So I think if you make a AIDS, HIV-resistant baby, this is a form of enhancement, right? You could argue. Should you do this or not? And I would think you don't know what you're doing, because while this receptor for the virus is therapeutically inactivated and cures people, right? So that's in clinical trials. This receptor also has another function. So what's not known that much, if you don't have this receptor, you're getting more susceptible to infection with another virus, coronavirus. So that's something which you have to consider. You don't know what you're doing. Another suggestion was, could we eliminate the ApoE4 allele, which is the most strongest risk factor for Alzheimer's, right? And it's pretty prevalent. So if you have ApoE4, your risk for Alzheimer goes up 30-fold or something. So it's really pretty bad. Shouldn't we eliminate this by editing this ApoE4? It's a one amino acid change to make it for ApoE3 and ApoE2, which is the protective alleles. Should you do that, right? And so eliminate this terrible gene from the population. What I just recently learned is so interesting. There's a tribe in South America. They have very high incidence of ApoE4 allele. 40–50% have the ApoE4 allele. Why? Because there's a selective advantage. Because they're more resistant to some parasite. That's what people believe. So what you do is these type of things. We have this diversity in our population to respond to changing environmental challenges.
Well, you talked about sickle cell disease, and there's sickle cell, and of course the connection to malaria.
Yeah, right. So sickle cell, of course, that is this mutation protects you against malaria. If you're heterozygous, if you're homozygous, you're very sick. So surely this is a terrible disease, but it has selective advantage. That's why this mutation is so prevalent in Africans. Yeah.
Yeah.
So a couple of points about, as I said in my presentation, we studied this matter of the difference between enhancement, therapy and enhancement, and it just isn't an easy distinction for a variety of reasons. And if you're interested in it, I'd suggest you look into that book, Beyond Therapy. It's actually, I think, the best thing we did as a council, and quite thoughtful, really. But let me just throw out a few things just that are quasi-scientific, but I think bear on the discussion. First of all, I mentioned designer babies, and I didn't say it, but there's also proposals and have been in the past for state-run eugenics programs. I wouldn't dismiss human cultures that are in the modern world that might go for that. I've talked to some scientists from Asia who say that their culture would be open to that. But I think we should get it out and make it clear. None of these large-scale enhancements are going to be easy to do. I think it's a myth that we're right on the cusp of significant designer babies. It's one thing maybe to alter a gene for susceptibility to HIV or ApoE4 or something, but the idea, what would people really want to do? They make more beautiful babies, more intelligent babies, babies that will live longer life. All those qualities in human nature are controlled by hundreds, maybe thousands of genes. And so far, for the most part, there's very, very little that you could intervene in with a single or even a small cluster of genes that would make a very large scale difference in that. And beyond that, we only know that certain genes work in certain contexts of other genes. And those genes will get reassorted, reshuffled with each generation. So in terms of making designer babies in a superior culture, that's not going to be an easy job. You agree with that, Rudy? And the primary reason is because of two basic principles. I tell my students, you got to learn these. If you learn nothing else from me, the concepts of polygenic inheritance and pleiotropy. Those two words that have big meaning in biology. One is that most traits are controlled by many genes, as I just said, and most genes affect many traits. So if you're going to go for something that you think this one gene is going to contribute a lot to, you're going to get a lot of other things you didn't bargain for. And that's why single gene genetic diseases often present medically as syndromes. You know, the person might have eyes too far apart, their webbed feet, they might have a heart defect. All these things because the single little tiny protein in the middle of it all is affecting many things. So I don't think these scenarios, large-scale scenarios, are very realistic in the current world and probably may never be. But so what we do about intermediate kinds of enhancement and private choice, as you say? Whose rights is it to decide? Well, I think there's some general principles we ought to not necessarily enforce, but take into our culture so that some of these things are not necessarily illegal, but unthinkable. And one of the problems we face as a modern world is that because historically we've had our cultures and we've had our traditions and they have been who we are. The very word ethics comes from the same root in Indo-European as ethnic. We are who we are because we are those people. And often, by the way, associated with the land they dwell on too. So ethics wasn't controversial the way it is in our modern world. And that's partly because of the advances of technology, but also because of the convergence of people from all over the world. And so we've sort of reduced in our society to the lowest common denominator for ethics, and that is don't impose suffering on other people. And suffering is sort of the greatest evil. But I think we have to be a little careful because the argument from suffering can be used to justify almost anything. There's more in the equation of human life than to combat against simply and directly against suffering. Now, I'm a doctor. I'm not in favor of suffering, and I'm not worshiping nature in the sense that I think nature is the perfect paradigm to be left alone. It's just that what we do in medicine doesn't, at least in good medicine, takes account of the whole person and the whole culture they're part of. And there are things that you could do that would relieve suffering that would in fact be, they would be evil actually. And I'm not in favor of those because I think the ends are not the only goal, the means to those ends also count for something. And that perspective honors human dignity in the process. I mean, if we said to the MIT undergraduate population, you know, we need just a couple of you to test this drug on, and what are your lives compared to the thousands of people it could help? Nobody would say, oh, that's okay. So we have to be careful before we think that way about developing human life as well. I'm personally not, I never really answered the question on embryo research, but I have big qualms about the use of human embryos in research. I think for both reasons of principle and reasons of prudence, I don't think it has a natural stopping point. Like I said in my presentation, one of my colleagues says that the definition of human life will eventually come through neuroscience, but if we really put Rudy to the test, he'd have a hard time saying when he thought human life had dignity, right, Rudy?
Yeah, well, it's a complex question.
And so what happens is, you know, one person gets off the train at one station, but somebody else goes on, and they say, well, that's your private culture that you got off earlier. But, you know, I'm in favor of relieving suffering by studying not just embryos, but fetuses. And, I mean, most people would find that, I think in the current world, find that abhorrent, but maybe not everybody. And then who gets to decide? Why can't they do research on why can't that guy, apart from the practical barriers of it, why can't he clone embryos, implant them, and harvest them for useful cells, tissues, and organs? How will we find an answer to that question? I struggled very deeply with this question when I was on the President's Council, because that was the most fundamental question we were converged to address. And I came up with what's a very unacceptable position in my own culture of university scientists. I decided that I was against embryo research. And I just want to add that although Rudy and I may have difference of opinion on that, I really respect Rudy's position on it because I, in the President's Council, came up with an idea for how you might get embryonic-like stem cells but not create embryos. And Rudy took the initiative here at MIT to do this project with mice and proved it could be done. And if the induced pluripotent stem cells hadn't come up, that might be the way it was done today. I think there are ways to bypass the ethical controversies, and wherever that's possible, I think we should do it that way in respect of human objections on this. That's a lot said in too short a time, but.
Yeah, no problem. So what I would like to do is we're going to soon transition to audience questions. I have one more question, but if you have questions from the audience, there are 2 mics located in the 2 aisles, so you guys can start lining up at the mics, and we're going to start taking questions from the audience. But I have one more question while you guys are getting there. And the question is that I was recently reading the Immortal Life of Henrietta Lacks, so the book about the discovery and development of HeLa cells. And one of the things that kind of stood out to me, it's kind of an interesting parallel story. One of the development of these cells and therapies and scientific techniques that were spawned by this immortal cell line, and then like the life of this family. And one of the things that stood out was that, like, along the way, it talks about how ethics in medical research was continuing to evolve. The things that were done when these cells were harvested would be unspeakable today, but that was common practice at that time. And so my question is that, how can the scientific and medical community move to a place where it's not reactive but proactive? I realize you guys are trying to do this, but it seems as if there's more people who are pushing the frontier, and it seems like usually what happens is we push the frontier too far, and then the public says, whoa, that was too far, we need to put a law up. We put a barrier up after we've gone too far. Is there a way to proactively put up something that will still, promote progress? It seems as if the history has been we go too far and then we regulate afterwards.
I mean, to me, it's reasonably straightforward. You should do your research under the permissible rules, right? I mean, of course, animal, whatever the rights what you have to follow. Do your fundamental research and get but be very transparent. And then you have the obligation to tell the public or whoever wants to listen, if you apply this technology to humans, that is a possible consequence, right? Positive or negative. So people are informed. So coming back to for me, the key border is really the application, the translation to medicine. And so the basic research, I think, is free, more or less. But you have the obligation to inform. Because if you don't do let's say if you think nobody would have done, let's say you opposed to genetic research or to gene manipulations, when should you have stopped that? When the DNA was discovered or when antibiotics were discovered? Because that was a basis for this, right? So there's no way to stop basic research, I think. It's a dead-end road. So you have to get the knowledge with all the potential, to my opinion, with all the potential of misuse or right use, and be aware of this and then make a decision, do you want to use it? So.
Yeah, well, I'm very largely in agreement with that, because I think basic research is the engine of transforming power. And as a physician, I know, for example, with CRISPR, there are probably way more now, but there are already 6,000. We'll soon have many thousands more, but we have already identified over 6,000 single gene diseases, 95% of which have no treatment whatsoever. That's an amazing body of suffering in our civilization that we need to address. I think one of the most important things about this current moment in this great story of the human drama of emerging scientific knowledge and cooperative civilization is that we, at this particular moment, have a chance to think ahead, prospectively. And one of the challenges of this, and Jennifer Doudna and I believe Rudy and all of us working together on this, I want to be very sure that the good that can come from this, the uncontroversial good, does not get stopped because it excites too much opposition, and in some cases reasonable opposition. And sometimes it's necessary to actually draw boundaries and say we won't do things just to keep the society coherent and to honor diversity of opinions. And sometimes you need to have protective barriers. I don't generally favor legal moratoria. It's much better if you can do this by social consensus. And yet there might be times when there need to be barriers. G.K. Chesterton has an interesting image where he says little boys are playing soccer on a field, but the field at the very borders of the field goes straight down to the 2,000 feet to the cliffs and the rocks below where the ocean's pounding. And so where are they playing soccer? Just in the inner 30 yards, maybe meters because it's Europe. Well, it was England, G.K. Chesterton, meters, yards. They're playing in the center of the field. But if you put a fence around the edge, safe, secure fence, they can play right to the borders. And so there's sometimes when you need to anticipate, draw boundaries, allow research clearly to some boundaries and not to others. But in the final end, what governs a society is going to be, it's a large, you start out with a worldview. Well, it's going to be large-scale questions of what the general ethos of the civilization is. And even in the matter of enhancement, I can think of some very good uses of enhancement. So for example, if I were a physician if I were a surgeon doing surgery on a child's eye, and I could take a beta blocker, which is a kind of interferes with well, I don't want to go into the biology of it. Basically, it'll steady the hand of the physician, the surgeon. If he's operating on an eye and it takes that for him to do a good job, even if it did something, sort of in a minor way, not destructive to that person, I would think that's a reasonable thing to do. Because in my mind, it's legitimate to give up something of your life for the good of something else. I think in some ways, I want to be used up in my life. I want every ounce of me to be used up in my life for the good. And when I die, it'll be like having drawn down the entire bank account of my biological existence. But you see, the point is, for the surgeon doing the surgery, the goal is good. But how do you know the goal's good? Like you started, what's the good? Well, that sets in the frame of a very large-scale understanding of what life is for, and what gives meaning, and what endows our life with dignity. And dignity is a hard word to define, but I think we all kind of know it. We certainly know love when we see it. And if we operated with the large-scale principle of love, C.S. Lewis once said that we should answer all of our problems with more love, not less love. I think that's a very good guideline.
All right, we're going to take some questions. We'll start here.
Hi, thank you for all your presentation and discussion. They're very wonderful. I'm a Chinese Christian, so I'm not so into that Chinese scientist about when life begins. So from the Christianity perspective, my question is, I have two questions. The first is, while we, the definition of joy and happiness, how do we define that? Because when people get aged, they have much more problems or trouble when they get aged. But a person of Alzheimer's, he might have less trouble, and he might have more happiness. So what if we ablate his Alzheimer's and also deprive him of his happiness? My second question is from the perspective of Christianity. So have you got my first question?
Well, I think let's try to stick to one question just to make sure everyone gets a question in. So pick your favorite question.
Okay, my favorite is C.S. Lewis once said, you are not a body, you are a soul with a body. So when people want to do the gene editing thing to make an internal life, do you really think we could achieve internal life in this body?
You're on, Rudy.
So did I understand the question?
I think the question was, could we get eternal life in this body?
An aging body.
Yeah, as Bill mentioned, there's some companies who claim they could do that. So I think, I believe this is nonsense. What we can do is, and I think that's a very serious and I think very useful way to do it. Can we really diminish the suffering during aging because one or the other organ fails in a terrible way? And I think there are ways regenerative medicine really gives you the potential to think about this and do this. I believe this is not the goal, would not be to make eternal life or to make you live for 200 years, but to make your last decade or whatever time it is more bearable and better. Because you have a clock in all your cells, and this clock runs down. And I don't think we are able to prevent that. But we can get when things go wrong in a given organ, be it a terrible disease like Parkinson's or Alzheimer's. Can we do something to make the progress slower of this disease or prevent it? I don't think it will necessarily increase your lifespan, but make it, the goal would be to make it more pleasant.
So next week, I'm debating a guy named Aubrey de Grey. You know this guy? He's a sort of futurist longevity guy, has a long beard. He was trained at Cambridge, and I'm doing something called a Chronicle Chats. The San Francisco Chronicle is having this in one of the major civic auditoriums in San Francisco. They're promising an audience of 600 to 900 people. And Aubrey de Grey, I debated him once years ago in front of 300 Silicon Valley engineers at Xerox PARC. It was a very high-level computer place. And he said to me, well, science is advancing so fast, if I can just live for 35 more years, I'm confident I will live to be 1,000. You ever heard this guy? So what do you think about it, Rudy? Are there any ways we can extend lifespans to 150, 180?
When you die, they cut off your head and freeze it, and then keep it for when people now are able. This is just really nonsense, right? This is really business and bad business.
Do you think any really radical life extension is possible, or do you think we'll pretty much
not with our biological body, but maybe need another body like putting this in electrons, right, and carbon-based. So it's a different type of life. So maybe that would be the solution. But
I think you should take my place in that debate.
It turns out, I've actually been genetically enhanced. We're all the same age. Yeah. There were side effects, though.
Oh, I thought you were older than I was. Yeah. Sorry.
There's side effects. I'm now black. So.
By the way, I want to tell you something. We had you weren't at this meeting, but one of our meetings with the CRISPR meetings at Berkeley, an African-American law professor from Columbia named Patricia Williams told us that she had an Asian student in the law school who married to another Asian student, and they decided to have a baby, and they decided, not for any medical reasons, but for reasons of social possibility, to seek a sperm donor, Caucasian, so that their child would not look as Asian, but would have the intermediate racial features, so that that child could have better social advantages in this culture. I think I'm shocked by that. I mean, I'm not surprised because I've seen this. I've had Asian students tell me their parents forced them to have the epicanthal fold, the upper eyelid cut so that they have rounder eyes. But I think, that's what I was saying a few minutes ago. Love does not look at people like that. And I don't think we should allow our society to go that way. And I don't know if you can make it illegal to do this sort of stuff, but I'm against it. I think it's not honoring to human life to do that. Either the surgery or the in vitro fertilization with a sperm donor on that front. That's a wrong approach, in my opinion.
Okay, let's take a question from here.
Hi, thanks. Okay. Hi, thanks for the talk. So considering a human rights perspective on medical treatment, and considering that as new medical technologies emerge, society starts to claim them as human rights, especially as they become cheaper and more available. I was wondering what your thoughts were on what gene editing technologies might eventually become a human right. People might start claiming everybody needs this. Like, I don't know, for example, people might start saying every child needs to be genetically checked and corrected, or everybody needs at least one extra set of organs grown for them just in case or something.
I didn't hear the question.
Yeah. So I see, I mean, I understand you correctly. With the genetic advances, would some bodies require that you are sequenced to predict your risk of a disease, or what?
Yeah. So just
so
for
that would be
gene
for
editing,
insurance is very important, right?
Yeah, for gene editing technologies
Yeah.
in general.
Yeah,
when will they become human?
I'm worried insurance will push for that. And I think that opens a lot of really complicated questions. Yes, I think sequencing will be probably soon in the order of $100 or something, right? Very soon you can do it sort of at home with a device. Yes, I think that will be the reality, will be that you will have your sequence and you will be able to genetically predict what your risk is for certain disease. Yes, I believe that will come.
I asked George Church if we'd have a $100 genome. He said it'll be free because the information will be so valuable. Yeah. Some company will pay you to do it.
Like Facebook, yeah.
But they're yeah.
Hopefully not them, maybe.
But you know, the professor at Stanford law professor, not a biologist, but the one that I quoted saying the line of human dignity will be determined by neuroscience. He's written a book about this, and he thinks that eventually all couples they'll create dozens of embryos, get them all sequenced before implanting them, and throw away the ones that have a 62% of depression and a 8% chance of dyslexia or whatever it is. See, so they'll filter embryos. I don't think he appreciates how difficult that's going to be, because most traits are controlled by many genes. And it's just the math doesn't work. And besides that, in vitro fertilization comes with definite medical disadvantages. There are risks associated. 60% higher rate of congenital birth defects with IVF. Multiple twins. There's now some worry that the IVF babies may have a slight, don't know, and don't worry about it if you're conceived this way, but they may have some medical risks that aren't in the normal population. And so we just don't know enough anyway to do this. So I don't think that's going to happen, but you see where it's going.
Hi, thanks for the talk. I have a question about the ethics of using, I guess chimeras, but in this case, instead of taking a human embryo or a human zygote and manipulating it or germline or whatever, we take, say, chimpanzee embryos or germlines or oocyte stem cells, and manipulate those, insert human genes into those. Presumably at some point, if you insert enough human genes into those, you'd get something approximating a human at some point. Maybe, I mean, unless you believe in that some soul juice is required for consciousness, possibly even a conscious, as in a human experience of consciousness kind of being. So what do you think about the prospects of something like that, and do you think it's ethical?
It's an interesting experiment, has been attempted, but it doesn't work. The reason is you have another genome. It is a mitochondrial genome. So the mitochondria are there's very few genes, but they're very important organelles, which make all the energy in your cell. So the egg brings all the mitochondria from your mother. You have your mother's mitochondria, not your father's mitochondria are actively destroyed, right, in the sperm. So now comes a problem. If you do it with interspecies, putting a human nucleus into a monkey, the mitochondria will not work, because many genes from the nucleus really have to match with the mitochondrial genes, and they're diverged. So the mitochondria will fail. So this type of nuclear transit will not work. Has been tried, it doesn't work. So you can be resting. Science at this point, you would have to replace all the mitochondria, make human mitochondria, then there's nothing monkey anymore left. So
I'm sorry, I just want to clarify if that's okay. I meant not a whole nucleus, but just take how that rabbit, for example, had the gene was replaced. Now it can glow in the dark. That was taking a gene from another organism and putting it in the rabbit. So in the same way, if you could take human genes and manipulate a chimpanzee's, insert them into a chimpanzee's gene, into a chimpanzee's genome,
So that can be done easily, and it's done all the time. So we could be putting human genes into mice for a certain reason that they function well. So that's, of course, you would expect. But maybe what you're asking is more fundamental. Could you put human genes which have been shown to be important for the difference of humans to primates? And some of these genes are beginning to be identified, right? So could you put those into primates or into other organisms and change their life? And there's an interesting experiment that's been done where people also from Stanford, they took a gene which is, actually, it wasn't, so I should set it up. So what this group figured out, there were certain regions which were deleted in humans of the genome, but present in primates and in mice, for example. So they took some of those and looked at this, and then they deleted this in mice. And the surprising result was the mice had 20% more neurons and had increased behavioral. And what really came out of this genetic region, apparently, when you didn't have it, you delayed neural differentiation. So neurons became later mature, which of course is a sign of humans get very late. Differentiation of neurons is very late, whereas in other animals who have to be ready to go in the wild after birth, humans are not. So I thought it was a very interesting experiment to try to define what are the genes which make humans different from, let's say, monkeys or from mice. Because our genome is 99% identical, right? So where are the differences? So this is the type of approach that's being done to look at this.
So there's a couple of interesting other things to say about that. So this has been called the human-Z, the idea that you've got this hybrid creature. And yes, as Rudy said with mice, there's also a study with a glial gene, I think, that the mice actually ran the maze considerably better. Yeah. And I mean, you know these are features have biological parameters to them. And so the question is, could you edge in, just let's say one gene per generation and edge a chimpanzee toward a human being? Well, part of it, I think, you can correct me if I'm wrong on this, Rudy, but you hear it all the time that the chimpanzee is, what, a 98.6 similarity in sequence or something like that. That doesn't mean that the chimp is 98.6 equivalent to a human being. It's just sequence of DNA. There's vast differences. For one thing, chimpanzees have another pair of chromosomes than humans. We have 46, they have 48, right? And there's lots of chromosomal rearrangements, and the rearrangements in the chromosomes make a difference in gene expression. And there's lots of little subtle differences that can't be quantified by the simple sequence. So my intuition on this, not being as deeply informed as my partner in presentation here, is that this is not nearly as easy as people think it might be. But it does raise interesting questions. You know, there's also proposals for, as I said in my presentation, for sort of reconstituting the infrastructural species that are our ancestors, and our cousin creatures like Neanderthals and Denisovans, and we now have it's a complete Neanderthal genome now, and so conceivably you could do that by cloning if you could perfect cloning. I mean, these are really amazingly difficult conversations. I don't know how you would adjudicate that. On the one hand, it would be kind of cruel to create just one. On the other hand, would you want to create enough that they'd mate? And in which case, what social position would they have? You know, somebody said, oh, they'd make great janitors. Well, that's disgusting. I don't think we should create a subclass of humanoids to be janitors. You can imagine, given human nature, a whole bunch of other things you'd use them for. I just think we have to be very careful, very respectful of what the order of being is. I mean, this is why I started my presentation by asking, well, what is nature? Where did it come from? I don't know. I mean, on one hand, you can make a purely evolutionary description of how nature got here, and I think there's a lot to be said for the material process of evolutionary unfolding, but it's not quite as simple as to say, well, it's all just, you know, tooth and claw, and it's all cruel, and it's all amoral, and we're just a coincidence within a chaos. I don't personally feel that way. I think there's a strange mystery of human meaning operating here, and I think there's some things we should be very, very reluctant to do, and included in those would be to vitiate those boundaries between other species and even our ancestral species.
Thank you.
So we're going to take these last 2 questions. We thank you all for saying we're like 18 minutes over, but the conversation's been good. So if you'll humor us, 2 quick questions and quick answers, maybe. We'll see.
You were first. Okay, thank you. Thank you for coming out here and having this conversation. I was wondering, in terms of the general concern over editing a human germline, do you think a CRISPR-Cas system that edits RNA instead of DNA, for example, with Cas13, do you think that that would kind of alleviate some of the concerns that we have over, oh, we're changing the human germline? And how do you think that would fit into the conversation that we had today, as well as the more, you know, general application of nucleic acid editing?
I mean, sure. RNA, of course, when you edit RNA, which is now coming sort of, is being established, it will be not permanent, right? RNA is, of course, short-lived. So the question is, how long would this last? And of course, whatever you want to do, you probably want to have it long-lasting. So I think it certainly will be not in the next generation. The same is for epigenetic modification, right? So you can change not the sequence, but you can change gene expression and gene activity by changing the chromatin configuration or the modification of the DNA, methylation, for example. This clearly will have a long-lasting effect, but will likely not go over the next generation. Will be erased. That's how epigenetics is erased and then reestablished. So I believe RNA has its applications, but probably not for this type of long-term genetic. At least I can't see this at this point.
Thank you.
Okay, last question.
So I'm assuming from history that no matter where you want to draw a line ethically, someone's going to want to cross it. So what do you do then? Whether it's on kind of the gross level, like Kim Jong-un decides that he can live forever if he experiments on political prisoners, or at the level of, say, MIT, where a professor argues there will be some incredible advantage to us if we cross it. How do you respond to that? What do you do?
So I mean, I have a very simple response for this. So this was the debate was when the debate was on cloning. Should you clone humans or not? And I thought it was really, Bill was involved in this debate in major, as he mentioned. So cloning, right? It's in a way you find this not acceptable for many reasons. It will be misused, right? Is that more than any other? It's illegal, because we're not allowed to do this. Still people will do this. It's a crime like any other. And it will not affect humanity. It will affect some individual. So is it different from going out and murdering someone? I'm not sure. It will not have an effect on the humanity if someone clones someone. It's very single, right? So I believe, so I don't have a huge concern for this, because you make a law and people will break the law and they should be punished. It's for everything. So I don't find this is very different from things like that, because you edited the germline. Well, it's not such a holy thing. If you edit the whole human germline for the futures, maybe that's something you would be concerned about. But for an individual, what's the difference to murder? I'm not sure, you have probably different.
I mean, you raise just an incredibly hard question, because with CRISPR, the powers of CRISPR-Cas9 and the possibilities for intervening in the genome, of not just humans but other species, you're talking not just about targeting recognized illnesses, and not just talking about trying to enhance human beings by somebody's scenario definition. And by the way, I don't think parents should have children by design. I feel like, you know, what's going to happen is the traits they wanted to bring out in their children, suppose they could actually affect them and make it happen. It'll be like the flavor of the month syndrome, that by the time those kids get to be 20, those traits won't be as valuable as they were 20 years before. I mean, it's a very strange thing. I think you're my own feeling is you're much better off to accept the children you have and love them. I have a handicapped child myself. She doesn't have a genetic problem. She was born, they made a mistake in birth, and she suffered very, very bad brain damage. They didn't get her heart beating until 9 minutes in her life. And I could have let her die, but I was not in favor of that because she was my baby. And so we asked them to try to do what they could, and she did. She lived. And I can tell you that it's nothing I ever would have asked for. But when you live a life where love is the central axis of your life, your whole family and your whole community is enriched by it. It's not, I don't think these utopian designs that people have are going to play out well. I think what we need to have is more love, not less love. But then when you get to we also want to have freedom and want to have rights, and let people do the lights of their own inspiration. But with CRISPR, we're talking about dangerous things. We're not just talking about therapies, we're talking about interventions in nature that could have large-scale externalities, effects on not just human beings, but the ecosystem. We're talking about potentials for bioterrorism that are just, I mean, I have a friend at Stanford who I mentioned to him that I was worried that the North Koreans might be working on biowarfare and chemical warfare. And he said, yeah, it worries me vastly more than their nuclear arsenal. And this guy's very well informed on these matters. So you can't just let everybody do what they want to do. There's now a history of people using synthetic biology to make viral genomes, very serious. There's a pox, a horsepox virus has been created, and it's probably provided now the instruction book for making smallpox. So at some level, you need to say no to people, where as far as possible within reasonable cultural parameters, you let people do what they do. By the way, I often get the question when I speak publicly, but what'll happen here? Won't the rich get all the advantage of this and the poor will be left behind? And my answer is this, that for the most part, at least for the foreseeable decades, it'll be the poor that are the lucky ones because they won't be experimented on by their parents. I think we should let life happen in these domains and love the people we're with. And the larger context, though, is if we do that, we can have a general consensus in our culture that just without having to make rules, laws, moratoriums, will get us general sentiment. You can feel with Rudy he's implicit, he says he doesn't believe in God, I don't believe in. I don't mean to say that superciliously, Rudy, but you obviously believe in good work, truth. You said you were after truth. That's what Jesus said he was, the way and the truth and the life. That truth is, if you, I'll tell you a story, just to end it. So I was very good friends with Arthur Kornberg, who was really probably, I don't know, arguably the greatest biochemist of the 20th century. Would you go with that? Arthur Kornberg discovered DNA polymerase. And he was my teacher, and I would go see him, every 3 or 4 months. Not regularly, but over the years I would see him often. And by sort of coincidence, because he was completely healthy, a couple of years ago I went in to see him, and he died 2 weeks later of an acute illness. So it was our last conversation, and somehow or another, I don't know how it happened, but we got into a conversation about death. And it was very interesting because although Arthur Kornberg was raised in an Orthodox Jewish community in New York, in Brooklyn, he had an evangelical Christian secretary. And every time I was in his office quite a few times when she'd come in and say, oh, there's this happening. And he'd say, oh, no, this is not going to work out. And she'd say, oh, Dr. Kornberg, don't worry about it. I'm praying about it. And it was always left him feeling very uncomfortable. But that was just, he really liked this secretary and that was his assistant. That was a really good thing. Anyway, we got in this conversation after one of these interventions by his assistant about death. And he said to me, well, you know, if I die and I go to heaven or I meet God, I guess he wasn't presuming he'd go to heaven. He said, if I die and I meet up with God and God says, Arthur, why didn't you believe in me? And he said, I will say to God, I'm a scientist. You didn't provide enough evidence. Okay, that's actually a famous quote. I think Sidney Hook or somebody said that. And I said to him, and you know, you have to understand the conversation. I wasn't being arrogant or anything. It was the kind of relationship we had. But I said to him, Arthur, that's not the way it's going to be at all. They said, if you die and you encounter God, you're going to start to apologize for not believing him. And God is going to say to you, Arthur, okay, maybe he didn't kind of see the full picture here, but you did believe in me. You did good in your life. You tried to do honorably. You sought the truth. You tried to help other people. And he was a wonderful teacher and a wonderful friend. And I think we need to be careful before we say what is and isn't God and what is and isn't true worship of God. God is love, according to the Gospels, according to the first letter of John. God is love. And it says perfect love casts out fear. Fear is driving a lot of troubles in our society. If we can love one another, those who love are of God at some level. So Rudy, I'm not willing to accept your self-designations here, but I do know this, that there's a mystery operating in our lives, that we all know our lives are about something, even those who say we're just a coincidence of chemistry. They feel it in their lives. I mean, I know Richard Dawkins, for example. I don't think Richard Dawkins thinks of his life as just robot vehicle, selfish genes in operation. It just doesn't ring true to what he says otherwise than when he's writing books and speaking publicly. I think we all know our lives mean something, and we all know, I think, implicitly that that meaning is somehow wrapped up in this word love. And we're all strangely seeking out what that word means and how to live it and how to promote it and how to give ourselves to it. And in that regard, if we do that, it's a long answer, but if we do that, I think we'll cover a multitude of problems that will never even arise. We could do lots of things with our biology that we don't do and that aren't even controversial. And we agree, we just don't do them. We don't talk about them. We could intervene in brains in ways we don't do, but it's not a controversy because we all know it's wrong. But those are framed by the larger social dialogue, and that's the way ethics works. We have to reestablish ourselves as the people of a certain kind of thought about what life is for, what it means, where it comes from, and what our individual and collective existence is about. That's what I think is the only answer, actually.
All right, well, I hope you all have enjoyed tonight's conversation. Before you leave, I just have a few short announcements, but while I do that, if you can please take a moment and fill out the rest of your survey card, and you can turn those in at the door as you leave. First, I'd like to invite everyone to a reception just outside in the hallway where we'll have food, and to continue this conversation. If you want to speak to these professors, I'm sure they'll be around for a little bit, and you can speak with them briefly. And finally, I just want to thank Dr. Hurlbut and Dr. Jaenisch and, of course, Dr. Bowie for their insights tonight and for moderating such a great discussion. Let's give our presenters and our moderator another round of applause.